It's just that so that we have a notion of what kind of conclusions we can take out from spatial transcriptomics data. And we'll end with some case studies, because I think it's relevant to understand what kind of biological studies can use this technology. So we can in general say that there are three kinds of transcriptomic studies. The bulk mRNA sequencing is probably the one that everybody has heard of, where basically we take a tissue sample, a plant sample, any kind of sample, and we extract the whole RNA from that sample, and then we do RNA-seq. This will give us the average gene expression in that tissue. However, sometimes it's more interesting to find out what the gene expression profile is of the different cells within that tissue, because a tissue will have many different kinds of cells, and of course they will have different transcriptional profiles that are being activated at that given moment. But now we can even add an extra layer of complexity with spatial transcriptomics. We can not only look at different cells, but we can also add spatial content, basically knowing where they are and which genes are they expressing. And this is what we're talking about today, spatial transcriptomics. So why study spatial transcriptomics instead of just the standard bulk RNA sequencing? These are some examples of areas of knowledge where spatial transcriptomics has been used. So for example, in the field of oncology, it has been used to study the tumor microenvironment. So how cells, the tumor cells and macrophages and other kinds of immune cells are interspersed within the tissue and are interacting with each other. In neuroscience, since the brain is an extremely complex tissue, it's very useful to use this kind of technology to create a neural atlas map, as in to find the transcriptional programs that are ongoing within the different areas of the brain. In developmental biology, this has been used to find cellular differentiation, morphogenesis processes that are ongoing during the different developmental stages. In immunology, this can be used to study the different immune cells that, for example, are infiltrating wound sites and to better understand inflammatory responses. And in botany, it has been used also to study the organogenesis process in flower buds, for example. So I think we can safely say that spatial transcriptomics really has a wide array of applications. It's just a matter of finding the best way to use it. So in terms of evolution, of course, spatial transcriptomics is a very recent technology. However, at least in the 70s, we already had what could be called a technique adjacent to spatial transcriptomics, and this is laser cut microsection. The idea behind this is that we would have a tissue and we would use a laser to cut a very small microsection of that tissue, and that area being cut would be the region of interest, so the one that we want to study, and that region of interest would be extracted the RNA and then do RNA sequencing, or do RT-PCR, for example. So basically, this would enable us to study only that region of interest instead of the complete tissue. Then this other technique called fluorescent in situ hybridization was developed. The idea behind this was using probes that hybridize to the targeted gene transcripts, producing a fluorescent output, which can then be read out. But this was only possible by using targeted genes. Then in 2013, this new technology called in situ sequencing was also developed. It basically used also targeted genes and a rolling circle amplification that would create a fluorescent output that was then read with a microscope. And this enabled to read the expression level of the targeted genes around the different parts of the tissue. But now we have this spatial transcriptomics that enables us to study the complete transcriptome without targeting specific genes. So let's dig in to how it works in theory. So I'm going to introduce the BMKGENE. This is a spatial transcriptomics chip that was developed in-house by BMKGENE. This chip has 8 active areas, 8 active capture areas, where basically each capture area is a different experiment. So let's just focus on one area. This area has 2 million spots. And in each of these spots, we'll have a well with a bead sitting on it. And the bead will be covered in these sequences. The sequence has a structure that you can see here on the slide. You can see that it has a poly-DT tail. This poly-DT tail is what enables the capture of our mRNA molecules, which have poly-A tails. Besides that, this sequence also has a UMI and a special barcode. As you can imagine, this special barcode will be different among all the 2 million spots in this capture area. It also has a read-one sequence, which will then be used for the library preparation for sequencing. So in practice, what happens with these chips is the following. We have the chip, we take a tissue section and we place it on top of the chip. After that, we permeabilize the tissue and the mRNA of the tissue is released. Using these beads, these beads will capture the mRNA and produce a cDNA molecule from it. And it's from this cDNA molecule that will then carry out the following steps and proceed with our spatial transcriptomics experiment. So as I said, this BMK chip was developed in-house. We started with the S1000 chip. This chip has 2 million spots, and the diameter of each spot is 2.5 micrometers. Then we also have a distance between the two spots, which is around 5 micrometers. And this distance is called the resolution of the technique. So it's the minimum distance where we can distinguish two different points. But very recently, we were able to actually upgrade this chip, and now we have released the S3000. This one has the double amount of spots. It has 4 million. And while the spots have the same diameter, the distance between them has decreased. So now our resolution is even better. The resolution is 3.5 micrometers. As you can imagine, by doing this and increasing the resolution, this means that using the same tissue and the same experimental workflow, we can get much more information in much more detail from the same experiment. So let's dig in a bit onto the features of this technology and how it impacts the experiments that are then carried out. The first one, which I think is extremely relevant, is the fact that its resolution is subcellular. What does that mean? It means since the distance between the spots is either 5 micrometers or 3.5, that distance is much smaller than the size of a cell. So we're talking about plant or mammalian cells that can be between 10 to 100 micrometers. So when we look at, for example, this tissue section here, where we have a different number of cells, it is stained with H&E so that we can see the cells. And then we overlay it with our chip. We can see that most cells will have multiple spots contributing to it. And most importantly, one spot will not be contributing and will not be contributing to multiple cells. This means that we can distinguish in a very good way the different cells within that tissue. This is particularly important when we compare to competitor technologies like this one below, where the distance is 100 micrometers. So this means that, for example, in the case that is circled here with the oval shape, we have a spot and we have, I think in this case, 3 cells that are contributing to that spot. That makes it a bit more difficult to then interpret the results, because if the 3 different adjacent cells are different and have different transcriptional profiles, that spot will have a mix of those profiles, so will not be as accurate. Another important feature is the fact that we can change the resolution level. And this is an image depicting how we do that. So when we have the 2 million spots and we have that resolution that can be 3.5 or 5 micrometers, that is the maximum resolution that we can get. But we may want to zoom out a bit of the image. It may be easier for us to try to not have the data so much detailed. And the way we can do this is we join adjacent spots in the form of an hexagon, as you see here in black, and we create a super spot. And this is resolution level that is above. Then if we want to further increase, we again join more adjacent spots to create the super spot in red, and so on and so forth, as you can see here. And we can reach up to resolution 13 resolution levels. So you would probably think that this is a bit weird. We talk so much about having a great resolution, but then we actually try to decrease it. So we have here an example of why this may be relevant. In this case, this was an hippocampus. And you can see with the H&E staining that we have already a clearly defined structure. And then we do the spatial transcriptomics data. And what we have here, the different colors, each color means a cluster of cells. So basically, the spots that are in orange represent spots that have a transcriptional profile that is very similar, probably the same kind of cells. And the ones in blue will be different kinds of cells, etc., etc. So when we go to a higher resolution, in this case, the 20 micrometer one on the right side of the slide, you can see that we have the defined layers, such as the ones that we see in the H&E staining. So the structure, the definition of the structure is there, but we kind of also have some noise. But when we go up a few levels in terms of resolution, for example, for the 35 micrometer resolution, now the definition is much better. So I think this depicts why it's important to have the possibility to zoom out and zoom in these experiments so that we can find the perfect resolution that fits our experiment. This BMK chip also has the advantage that it's compatible with multiple sequencing platforms. So although our experiments are customarily performed in Illumina, so this means we use short reads, it is also possible to carry out these experiments in nanopore. And this is because after we perform the mRNA capture and cDNA synthesis, we get a complete cDNA molecule, so the full-length transcript. So then we can choose to, instead of using Illumina, we can choose to prepare a nanopore library and then sequence it in nanopore. And what we have here in the slide is basically one case study where the same sample was analyzed in both Illumina and nanopore. And you can see that after clustering clustering the spots, we can see that the clustering is very similar between Illumina and nanopore, thus validating the use of nanopore for this spatial transcriptomic technology. And finally, and I think this is likely the most relevant feature for spatial transcriptomics method, is the fact that we can split the transcriptomic data at the level of cells and not only spots. This means that we can use fluorescent staining plus H&E staining plus the sequencing data that we obtain. We integrate all that together and we can assign the spots to particular cells. So instead of analyzing spots, we analyze cell boundaries. And let's look a bit in more detail to how this is done. So first we start by having our section, and we do a staining of the nuclei with fluorescence. We read out that fluorescent image, and then we use a watershed algorithm that will predict the cell boundaries of each cell in the tissue. Then we perform H&E staining, and we correct the cell boundaries based on this image. Finally, we sequence the reads of all the spots, and then we assign each spot, we assign the spots that are contributing to each cell in the image. So in the end, this means that we get an image just like this one that you can see on the bottom corner. This is an example of a testis sample. And as you can see, the cell boundaries are clearly defined, and this means that we can then cluster the different cell types within the testis. And even using annotation, cellular annotation, based on their transcriptional profiles and compare them to public databases, we can say, for example, that the ones in blue are likely spermatocytes, while the ones in, the ones in green are likely spermatocytes, the ones in blue are likely Sertoli cells, etc., etc. So this is a much more informative way and with much more biological meaning to study spatial transcriptomics. So I would just like to highlight that when doing a spatial transcriptomics experiment, we do the clustering, as I mentioned previously. So we get images like this, where we get each color or each cluster corresponding to a specific cell type, or that's what we would expect, or at least a cell type that is undergoing a different transcriptional program. And when we do with the samples such as the mouse here, we can then compare it to our previous knowledge of the mouse brain, where we know the anatomical regions, and also previous knowledge on functionality or regional subtypes of cells. And we expect to find something like this, where the transcriptional division of the cells is very similar to the anatomical structure and also to the regional subtypes. So this is not only a validation of the spatial transcriptomics technology, but it also means that we can then use the data that we have here for each cellular cluster and try to find out more what is happening in these cells. So what genes are being upregulated, what cellular process may be ongoing in these cells. So now let's take a kind of a deep dive for the experimental workflow from sample to data collection. So let's start with the sample. We can have a sample, for example, an animal tissue or a plant tissue, any kind of sample, and we need to embed it in an OCT cassette. Since there are different sample types, this procedure is kind of straightforward, but there may be some variation depending on the different sample types. So we have this document that has a sample preparation guidelines to help to understand what is the best procedure. After embedding in the OCT, that is frozen and then placed in the -80 storage. Then it is put in a cryostat and we start cutting the OCT cassette. This is called cryosectioning, and we get those sections. We then do H&E staining to find out at which part of the block is the structure that we want to analyze. So for example, if we have an embryo in the OCT cassette, we need to make sure that we are cutting in the right area where the structure that we want to analyze are there. So we need to cut more or cut less to find the correct area of the OCT cassette. After that, we need to make sure that RNA that we can extract from these sections has a high integrity. And the way we do this is we obtain 10 to 15 sections from this cassette. We extract the bulk RNA from those sections, and then we evaluate the integrity of the RNA. And this is using the RIN number, which is a very standard measurement, and we expect it to be above 7. This is extremely important because if the RIN number is low, this tells us that RNA is fragmented, and in that case, it's not very advisable to continue to the next steps because likely the formal experiment will not go so well because fragmented RNA is not easily captured. So when this happens, we actually advise to obtain a new sample that has an RNA that has a high integrity. After this, we need to find the optimal conditions to permeabilize the tissue in question. Since this technique can be used, as I said, with multiple kinds of samples, there are samples that require a very long time of permeabilization for the mRNA to be released. For example, plant samples have that cell wall that is difficult to open. So this means that the tissue permeabilization time will likely be longer. But when we compare that with, for example, brain or samples that are softer, probably the permeabilization time should be shorter. So every time we have a new tissue, we do this tissue permeabilization optimization. This means that we place the tissue in the ship and we apply the permeabilization reagents and we test out different incubation times to find the one that is optimal. And what I mean by optimal time is we need to achieve the best mRNA release that we can so that we can capture it, but we also don't want it to be so long that mRNA starts to diffuse. Because if the mRNA diffuses, it will go to the adjacent spots. And of course, this will result in a data set that is not so perfect. So after these two, we have all the conditions gathered to then carry out the formal experiment. So in this formal experiment, we have the S1000 or the S3000 chip, which is depicted here. You can see how it looks. We take a tissue section and we place it on the chip. We then have H&E staining and fluorescent staining, and we obtain those images, we capture. Then we permeabilize the tissue, we capture the mRNA, and we synthesize the corresponding cDNA molecule, which, as you remember, that cDNA molecule will have the special barcode from each of the beads and also a UMI. After that, we have that cDNA molecule. We just need to prepare an Illumina library. So the library will have a structure like this. What matters the most here is that it will have the special barcode and the UMI, which are really important so that we can analyze the data. After having the library, we sequence it in Illumina NovaSeq. For this setting, we use paired and reads that have 150 base pairs of length. And the data output will actually vary between experiments. So the S1000 chip, since it has only 2 million spots, requires less data than the one that has 4 million spots. Also, if we want to do cell segmentation in the project, we require more data so that we can have a precise cell segmentation procedure. So this data recommendation may vary depending on the project. And after that, we have a data analysis. So basically all the bioinformatic analysis that we can get out of the data. And now let's take a little bit of look at the bioinformatics part. So it starts with raw data. Of course, we do a quality control to ensure that the sequencing procedure went correctly, and there are a list of parameters to analyze quality control in the context of spatial transcriptomics. And then for all the reads, we need to identify the barcodes and the UMIs in those reads. We also need to analyze, to align the reads to the reference gene. This is how we can say that we can assign each read to a particular gene. And then we do data statistics. And this can be done at a multi-level or a cell segmentation level. So let's go back again to talk about that multi-level resolution. When we are at the biggest resolution possible, so the 5 micrometers, for example, we do statistics for this part. But then we zoom out a bit and we do statistics for the subspot level 2 with 10 micrometers, and etc. and etc. And we do this with 13 levels of resolution. We also do the statistics, instead of applying it spot by spot, we apply it cell by cell. And the basis of all this is gene expression matrix. So this is a very standard format that is used both in spatial transcriptomics experiments, but also in single cell ones. And the data format has this structure where we have, in the lines, we have all the genes identified in the experiment. And in the columns, we have all the spots identified in the experiment, or in this case, all the cells, if we're analyzing spots or cells. And for example, looking at this gene expression matrix here, of course, it's a very simple example. We can see that in cell 1, the expression level of gene 1 is 3. And so this is the idea of this gene expression matrix. This is a very standard format and can be used for all the downstream analysis. So our standard package of bioinformatics includes a very extensive downstream analysis, and it's mostly focused on clustering of these spots and cells. And I'll go over a bit to see what kind of information we can get out of it. So we have quality control, and then we can start by analyzing sample by sample. That is, not comparing different samples, but only looking at one sample by itself. And this is what we call the inner sample analysis. So first we start by detecting the UMIs and the genes in the tissue, and we get an image like this, where we get where we know the number of genes that was detected in each spot. Then we do spot clustering. So just a reminder, this means that we look at all the spots and we find the ones that have similar transcriptional profiles, and we cluster them together. And we can analyze them or look at them using these reduced dimensionality graphs, such as this one on the right. And we can see how the different clusters are closer together or not. Or we can also look at the tissue and see how the different clusters are interspersed within the tissue. So as you can expect, for example, the cluster in orange is very close together on the outer layer of the tissue, and then we have a layer of blue, etc., etc. This again goes to that idea that the transcriptional response in the clusters of transcriptional similar cells should be similar to structural information within the tissue. Then we do an analysis of the differential expression between cluster genes. And this is what we can see here on the heat map, or basically each cluster has at least a number of genes that will have a significantly different expression in that cluster when compared to all the other clusters. And it is based on this that we can identify the marker genes. So these marker genes can be used to identify specific cluster. Basically, their expression should be extremely high when compared to other clusters. For example, when we look at here the gene VXN, it should be a marker gene, and we can see its expression around the tissue type. And we can see that it's mostly concentrated within a specific area. Then we do functional analysis of these differential expressions genes. And this is just to tell us if there is a particular cellular process that is ongoing and it's more relevant in one cluster or another, or if there's some specific metabolic pathways that are being highly expressed, etc. After this inner sample analysis, we go to the intergroup analysis. And this may be useful when we have projects that, for example, compare two conditions. It could be a condition where the sample is not treated with the drug and the other one is treated, or it can compare different developmental stages, just two examples. So in the intergroup analysis, what we do is we get all the data from all the samples and we combine them together, and then we recluster everything. So for example, here in this slide, we can see that we had a posterior condition and an anterior condition. And we have the different clusters between the two samples. And again, we can look at them using reduced dimensionality graphs, or we can also look at them just by looking at the tissue. Then the remaining of the procedure is similar. We do differential gene expression analysis between the different clusters, so we can have this heat map and find the differentially expressed genes. And we can also identify marker genes for each of the clusters. We also do functional analysis of these differentially expressed genes. So this, all that I've been saying previously about bioinformatic analysis is within our standard bioinformatic report. But outside of this, I would just like to highlight that in our website, we have these two tools that can be downloaded for free. I think the most important thing about these tools is that they have a graphical interface, which you can see right here. It doesn't require any kind of coding. It's just importing data and clicking buttons. And it's extremely useful to analyze these data sets. So the first one is the BST viewer, and it enables us to input the H&E image and the sequencing data, and to then do clustering of the spots. And we can do it at those different resolution levels that I've been mentioning. We can also select specific regions of interest and just analyze those ones. And this can be important if the tissue is very big and we just want to analyze a specific part, or even if we have more than one specimen, or basically more than one sample in the same section, and then we can separate those. We also developed the BST cell viewer software, and this is the one that is used to do cell splitting. So in those projects that we want to analyze by cell and not by spot, we input the data, and it does automatic cell splitting. So basically, it automatically finds the cell boundaries and attributes the information. Or it also enables the user to correct the cell splitting manually. In some cases, if the user thinks that some cell boundaries are not correct and should be corrected. So that is it about bioinformatics. I would just like to take this last time of this webinar to talk about some case studies and basically look at the utility of this spatial transcriptomics technology. So this first demo case was generated using a mouse embryo, and it was with the S3000 chip. So that chip that is upgraded, that has more spots, and that resolution is 3.5 micrometers. Using this chip and sequencing in Illumina Novasec, obtaining 1 terabyte of data, so a very large amount in this case, it was possible to analyze the performance on different resolutions. And I'm just going to focus on the 6 micrometer resolution because that's smaller and that's more informative in that case, that we can obtain around a bit over 800 genes per spot, which I think is extremely relevant considering the small size of 6 micrometers. When looking at cell segmentation in this section, it was possible to detect more than 200K cells, and the median genes detected per cell were around 1.7K per cell, which is also a very good number. So in practice, what we can see here is looking at the H&E staining and then at the spot clustering. So in this case, the cell clustering, we can see that we can get extremely detailed views of the different kinds of cells that are present there and their dispersion. I also bring here an example that was published very recently in Cancer Cell, and that used this spatial transcriptomics S1000 chip. In this case, the authors were interested in acromelanoma. This is a cancer type that appears in the skin of the hands and feet. And they were interested in trying to study the process of invasion, of tumor invasion. So this large cohort analyzed 2 kinds of samples. One was samples that are in early stage, still in situ, and that have limited understanding, so that they haven't been studied that much. And the other kind of sample are the later stage invasive melanoma, and that have very poor prognosis. So this was a multi-omic study. It's used a very large data set and also very different techniques. So I'm just going to focus on some key points here. So using these 3 omics approaches, exome sequencing and bulk RNA, the authors were able to pinpoint that there are these types of tumor-associated macrophages that infiltrate the tumor and may trigger the transition from epithelial to mesenchymal. And this transition is important for invasiveness. And so basically these TAMs may lead to a poorer prognosis. Building on this conclusion and then studying single-cell RNA sets of these tumor types, the authors were then able to specify a specific subset of macrophages, which are highlighted here, and find that these macrophages are positively correlated with these epithelial mesenchymal transition scores. So basically, they are correlated with cells that can then do this transition and become invasive. So of course, being able to use spatial means that we can now study the interactions between these macrophages and the tumor cells. So this is what the authors did. They used the BMKGENE S1000. They analyzed 10 samples, and samples were divided in 2 types. The ones on the left here are the ones that have a high invasiveness potential. As you can see here, there is a tumor region. And the ones on the right are the ones with low invasiveness potential. The analysis basically used some bioinformatic analysis that I have already described here. So cell segmentation, marker gene identification, cell type annotation, and then also cell-to-cell interactions. As you can see in these images, this kind of spatial transcriptomics enable to find the to very clearly define the different cell types in these tumors, in these samples, skin samples. And for example, looking at the epidermis, it's possible here to define the 3 strata of epidermis of different cell types. The tumor region is also very well defined. And when looking for melanoma markers within this tumor region, you can see, for example, M-LANA and CDH1, they are expressed in the tumor as expected. So going into the things that are not so expected or not so, the things that were actually being studied here is the fact that authors were able, to find high number of these specific kinds of macrophages in the C3 subtype tumors. And most importantly, they found that there is a closer physical distance between these ApoE macrophages and the tumor cells that have a high invasive potential, or the EMT tumor cells. So this direct evidence that the cells are physically close, it's very important to confirm that actually this may be a mechanism behind it. They also analyzed the receptor and ligands that were being expressed within these cells, and found that this interaction may be mediated by some signaling pathways, which I don't describe here in detail, but it's extremely important to then further study these signaling pathways and how they are important in this invasiveness process. So you can see here that having spatial content can be extremely relevant to find these direct interactions between different cell types. And finally, the last case study uses a very different sample type. It's focused on tomatocallis, so a plant sample. And the idea here was to study how it regenerates, so a regeneration process. The authors obtained a tomatocallis culture and then analyzed it using spatial transcriptomics techniques, and those included the BMKGENE OS1000. So starting by those standard analyses, looking at this tomatocallis sample, it was possible to cluster the cells, as you can see here. And in some cases, these clusters were able to be, were annotated as specific cell types, and in other cases, they were not, maybe because those cell types were not present in the public databases. So you can see, for example, some cells were classified as inner callus or vascular tissue, etc., etc. And then looking at those clusters that were correctly annotated, it was also possible to find those marker genes. So for example, in this last cluster called outgrowth shoot, you can see that we have 3 marker genes that have an extremely high expression that is only present in that cluster, which is expected. But this being a study focused on regeneration, the authors were specifically interested in this cellular type called shoot primordial. So they took an in-depth look at that cellular type. First, they were able to see that these cells, these shoot primordial cells, had a high expression of genes involved in multiple signaling pathways, namely including these molecules of auxin, ethylene, salicylic acid, etc. So this was already important to try to understand how these signaling molecules may be contributing to the organogenesis process. But furthermore, they also found out that these shoot primordial cells, which here in this image are depicted in red, are enriched by cells that are upregulating photosynthesis genes. So it seems that there is an interaction between shoot primordial cells and cells that are actively photosynthesizing. Building on this, the authors then performed histological examinations and were able to confirm that those cells around the shoot primordial ones are actually chlorenchyma cells. And considering the role between chlorochemia and light induction, this was a conclusion that it was possible to build on to then further study how light induction promotes callus regeneration. And this again is mediated by the interaction by these two kinds of cells. And again, just to emphasize, this is only possible when we look at spatial content and at physical interaction between these kinds of cells. So that is all that I have to talk today about spatial. I would just like to highlight that not only at BMKGENE we do spatial transcriptomics, but we also do many other kinds of studies. We can help you with transcriptomics, metagenomics, genomic and populational studies, etc. So if you have any question, I'm happy to answer.
Yes. Thank you, Anna, for the excellent presentation. Very good. And now we have some questions actually that we are planning to answer. So the first question is the following. I work with very small specimens, such as mouse eye, which are much smaller than the area of your chip. Can I put multiple specimens in one block to just use one chip?
Yeah, so when we have multiple, when we have small samples, we actually advise to put more than one sample in the same block. The reason is if the sample is very small and we just put one in the block and then we cut it, that means that only a very small portion of the section will be covered by tissue. And we advise that at least 40% of the section is covered by tissue. This is because if it's less than that, we basically have less RNA to capture. So then we have less cDNA that is produced. And of course, this means that the whole process will not work very well. And also it will be a waste in terms of using a chip and then just analyzing a very small sample. So for this reason, when we have small samples, we advise to put more than one sample in the same block. And in those cases, we just need to figure, depending on the sample type, and we can discuss, depending on the projects, that they are in the same Z-plane, meaning we have one sample here and another one here, and then when we cut them, they are present in the same cut. So this is something that can be discussed case by case, but it's possible.
The second question is, why can't your ST chip analyze FFPE samples?
Yeah, so the reason is FFPE samples, so the paraffin ones, have fragmented RNA. And this means that it's not easily captured using poly-A methods, which is the method that we are using here. However, since FFPE samples are extremely used, especially by hospitals, the 10x, we do have a 10x genomics service that we can also provide, and that 10x genomics can use FFPE samples. The resolution is not as good as this one, but it's possible for FFPE samples because it doesn't use a poly-A method. So in that case, it can overcome the problem of the RNA being fragmented.
Perfect. And the third question is, I work with a CDX model where we inject a human tumor in a mouse. If we want to analyze the interaction between both human and mouse cells, it is possible to do this?
So this is when we have two species types in the same sample. When that happens, we can do the analysis. It's possible. We just do two separate analyses. So with CDX models, we have mouse and human. So we first do an analysis against the mouse with the mouse genome, and then another one against the human genome. And that means that we are able to separate those cell types. We also need to ensure that, for example, a cell, when we analyze, for example, against the mouse genome, we need to remove all the human cells from the data set. And the way we do that is we have a filtering step where we remove cells that have a very high mapping ratio against human, and vice versa.
Perfect. Another question, do you have experience with ST with different plant types?
Yes, yes. So it depends. Plant cells are samples are a bit more tricky, like not so straightforward. But we do have experience with plant tissues. In general, it's better if they are fresh. This is something that our experience has told us that really it's not easy to analyze old plant samples, and that is also why we provide that guidelines document to find the best way to embed and preserve those plant samples. But yes, we do have experience.
Nice. We also have another question. I'm working with different plants. What about the spatial transcriptomics in plant samples like leaf, stem, tissues with cells walls and different from plant callus? Think you
Yeah.
are, yeah.
It's similar to the previous one. So basically, it depends on the plant tissue. So depending on your plant species, we can analyze it and see our internal experience and how it works. I'm also just highlighting that we have this tissue optimization step. So this means that, for example, plant stem and plant leaf will have different incubation times during the permeabilization. So during that step, we'll optimize it for your tissue. But of course, this is something we analyze always case by case. So I would be happy to discuss your samples and see what our experience is with it.
Perfect. And then we have a last question. What is the different price between the S1000 and the S3000 chips? And Emma already answered to that question. So we will introduce our sales representative by email later, so they can get all the information about that as well. I think that's it. Yeah. Yes, thank you very much, Anna, for your presentation and answers. Yeah, I'm pretty sure we all got very good insights from today's webinar. Thank you very much for that.
Thank you very much, everybody. Bye.
Yeah, sure. And as we conclude today's session, yeah, we're grateful for your participation. Please look out for the follow-up email with the webinar recording and check out our social media page for future webinars. And thank you again. Have a good day.